that it's all up and running, we are going to continue. We might finish this PowerPoint, I don't know. With the adaptive immune system. Just kind of as a recall from last time, I'm going to turn some of this lights lower. Adaptive immunity, this is where it can recognize very specific invaders. Once it recognizes those invaders, we can activate our cells. They can go get rid of those very specific invaders. They can make clones of themselves to help get rid of those invaders, and then we can make memory cells so that the next time you get infected with these things, your body remembers how to get rid of them and you have a faster response. But the adaptive immune system itself, it does have two branches to it. There's the humoral branch, which we got through on Tuesday, which involves the B cells because they mature in the red bone marrow. Once they recognize with those receptors, with the little hands, the very specific things they're meant to recognize, they can turn into plasma cells, and the plasma cell's job is to turn out tons and tons of antibodies. If they didn't turn into a plasma cell, those activated B cells became memory cells. And when they recognize that same foreign invader again, they can make plasma cells a lot faster, they can churn out antibodies a lot faster, and hopefully they can get rid of whatever it is that got in your body a whole lot faster. Now, the humoral branch is there to recognize extracellular pathogens, so things that are in the body but that are not inside of our own cells. So today we're going to get to the cell-mediated branch, and depending on how far we get, we'll talk also a little bit, they do chat with each other. So like the humoral branch and the cell-mediated do work with each other, and when the two branches work together, of course, it's always better if you work together. We're going to talk first just, what can the cell-mediated branch do all by itself? So the cell-mediated branch has anything that has to do with T cells. So there were B lymphocytes or B cells, and T lymphocytes or T cells, just shorten the word. Although T cells do mature in the thymus, they were made in the red bone marrow. Just the B cells stayed there and matured. The T cells head off to the thymus, and that's where they mature. That's where they get their receptors to recognize very specific things. Once they've matured, they kind of migrate around. They kind of patrol the body looking for foreign invaders. We can find them in the lymph, we can find them in the blood, we can find them in the lymph nodes, the spleen, and then in Peyer's patches. Peyer's patches are, kind of briefly talked about it like that MALT, that mucosa-associated lymphoid tissue. It's like tissue areas where you have high concentrations of B cells and T cells. It's in areas where you're going to have a highest risk of encountering some type of foreign object, and your digestive tract is a huge area where you may encounter foreign objects. And so the Peyer's patches is one of those types of groups of tissues in our digestive tract. And the T cell's job is to recognize infected intracellular organisms. So there are viruses that get into our body that they can't reproduce on their own. They have to get inside of our cells. They have to hijack our cells, and then our cells become virus-producing factories. But our immune system has to be able to recognize one of our cells and be like, oh, you have something foreign in you. You have a virus in you, or you have a bacteria that's hiding out in you, and I need to kill you because you're infected. And so we have T cells that can recognize the fact that there are these intracellular foreign objects. So T cells are attacking invaders inside, B cells are attacking invaders on the outside. Now, T cells also have T cell receptors on the outside, but they do look a little different than a B cell receptor. I don't know if I want my pointer, if I want just pictures. So if you remember from last class, if you had a normal B cell, it had B cell receptors all over it that look like little Ys. That was your normal B cell. A T cell, still a lymphocyte, still cell. It's also going to have receptors all over it. They just don't look quite like a Y. They look like two little sticks that stick out. They don't have kind of those arms with the hands that are looking for something. So here is our T cell. And it has like two little sticks on it. Now, it recognizes that this is one of your cells that is infected with a virus because your cell is presenting that virus. when you have an infected cell, some of that foreign object is presented on the outside. So this is a green virus. I'm just going color coding. I'm like, this is a green virus, and some of that green part of that virus is presented on the outside. So a T cell can't actually see the virus. The only reason it knows that a virus is there is that the cell that's infected is showing that it's infected. And the T cell has to be able to recognize it. It can't see inside of it. It has to be shown that it's there. So T cell receptors only bind epitopes. Those are foreign objects that can be recognized. They're the little things on outside of antigens that are associated with an MHC. Now, if you recall, I'm going to erase some of this after I write it up. There were MHC class 1 antigens and MHC class 2 antigens. Whether you have to look back at your notes or not, do you remember what cells carry an MHC class 1 antigen? You may have to look back at your notes.
All nucleated cells.
Yes, all nucleated cells. Which pretty much means like every single cell in your entire body, except red blood cells. So all nucleated cells do have an MHC antigen. That just means every cell in your body has the ability to present foreign objects to T cells to recognize. MHC class 2 antigens, what cells were those on? There was like 3 of them. Yep. They're on antigen-presenting cells, and that can include like macrophages. Those are big eating cells, dendritic cells. I was like, and even B cells can present antigens. I can't spell dendritic, can I? Dendritic. So these are specialized cells that their entire job is to eat things and then present it to T cells. So T cells can't recognize foreign infected objects. They have to be always presented it on one of these MHC antigens, always. So it's this like unique thing where a B cell could normally just be trolling around looking for foreign objects with little hands and attaching to it. T cells have to be shown that there is a foreign object through one of these MHC antigens. Again, all of your cells have an MHC antigen, so they can always show it if you have a virus or a bacteria or something else that got inside one of your cells. Now, there's more than one type of T cell. It's like a lot on one slide. It's like some faux pas of PowerPoints. So it's the reason why I didn't write out the word T cell yet, just like I wrote out B cells. There are 3 types of T cells. B cells are just B cells. T cells, a little different. There are cytotoxic T cells. We usually just call them big T with a little c. So it's a T cell, but it's a cytotoxic T cell. There are T cells that are regulatory T cells and T cells that are helper T cells. I didn't go in my order. Now, although there are 3 T cells, they're in 2 different groups, just based on what their T cell receptor looks like. T helper cells and T regulatory cells are called, just because you may see the word, I don't ask about it, but you may see the word somewhere, CD4 and CD8. Just has to do with what that little receptor, that little stick receptor looks like. Now, based on the name of cytotoxic T lymphocyte or cytotoxic T cell, cytotoxic T cells, based on their name, cytotoxic, they create toxins to kill cells. Their whole job is to kill. They are going to see an infected cell because they were presented something foreign, and then they're going to kill that cell. That's their job. They are killing machines. Helper T cells, their job is really to help activate the immune system a little bit more. They can activate the job of the cytotoxic cells. They can also, I don't want to get there yet, also help activate the humoral branch. Their job is to help. They just kind of help everywhere. They help those cytotoxic T cells become better killers, and they can also help activate the humoral branch. So they can do anything that's there to help. Regulatory T cells is there to suppress the immune system. Your cytotoxic T cells are so good at killing that sometimes they kill healthy cells. And so the job of a regulatory T cell is to kind of keep them under, under control, in check. And a lot of times people that have immune disorders, autoimmune disorders, where their immune system is attacking their own cells, it's because the regulatory T cells are not doing their job. So, which is, now that we know this, we can try to figure out how can we boost the regulatory T cells in those patients. So their job is just to suppress the immune response. Now, any of these can turn into a memory cell. We can have memory regulatory cells, we can have memory helper T cells, and we can have memory cytotoxic cells. Now, bigger picture, and I'll kind of break it up a little bit. So this is just showing where do we get our T cells. And we don't talk a lot about the regulatory T cells other than, they are CD4, and they suppress the immune response. But we have your immature immune cell. It's made in the red bone marrow, but it goes to the thymus to mature. That's where we get our T cells. They're either going to be a CD4 or they're going to be a CD8. The CD8 is where we get our cytotoxic cells. The CD4 is where we get our helper T cells and our regulatory T cells. Again, you see these names and numbers and letters. Now, what kind of cells can activate them? Cytotoxic cells are activated by anything on the MHC1. So any cell in your entire body can activate a cytotoxic cell. Yeah, any cell except red blood cells. But not every cell can activate these helper cells and the regulatory cells. They only get activated to do something when they're presented with an MHC2. And those are on those antigen-presenting cells. So here's an antigen-presenting cell. It's an example, a dendritic cell. Again, it's called dendritic because it's had these long extensions off of it. It's an antigen-presenting cell, so it has that class 2 antigen on it. And once this T cell recognizes that little red rectangle, it activates it. And it starts to go through clones, or clonal expansion. It starts to make more clones of itself. Because again, if you're going to fight a war, it's best to have a big army. And so it starts to make more of itself so that it can go do whatever it needs to do, whether it's regulation or whether it's helping the cytotoxic cells or whether it's helping the humoral branch. And I'm like, but it's activated just by that MHC2. The cytotoxic cells, they're activated by anything on that class 1. Which again, all of your cells have a class 1, and so any of your cells can activate a cytotoxic cell. Again, once it recognizes something, and I'm like, it becomes activated, it goes through clonal expansion, makes lots of itself, and they go off and go kill. Any of these cells can make memory cells of themselves. Now, an antigen-presenting cell, I have it listed both. An antigen-presenting cell does have class 2 antigens on it, but it's still a cell, and it still has a nucleus, which means it does also have an MHC1. All cells in the body have an MHC1 antigen, and antigen-presenting cells have, because they're a cell, they have an MHC1, but they also have an MHC2. They have 2 different types of presenting antigens. So antigen-presenting cells can activate your T helper cells, your regulatory T cells, and they can also activate cytotoxic cells. Again, if you're infected with something, it's best to wake up everything so that everything is working all together. Now, what helps with all of this is, again, if your cells chat with each other. And so there are some ways that they can start to chat and interact with each other. Because again, they can't call each other and talk to each other and tell them, you know, what's all going on and who needs to do what. So they use different types of signals. Usually they're all types of proteins. One of the first signals is an interleukin. It's just a chemical that's released that goes between white blood cells. Any white blood cells. It doesn't even have to be lymphocytes. The word inter means between. Leukin is for white. So the word means, you know, between white blood cells. It allows white blood cells to chat. And you'll actually see IL in a couple of my pictures. It just means, again, that's a chemical that you can see. It's doing stuff. It's going between stuff. It just allows white blood cells to chat. It could be they're chatting amongst themselves, whether it's a T cell or B cell. These are all white blood cells. Or it could also be chatting with different types of macrophages. The big eating cells. They're still white blood cells. It's great if you can recruit them too. An interferon is an antiviral. I was like, it's always a protein, but protein, communication. And it's actually in our second line of defense. So if you watch the video, read through it, interferons do allow cells to chat with each other. So if you have two cells, and this one becomes infected with a virus, there's my virus. This infected cell can actually send signals to that cell. That would be your interferon. And it can tell that cell, you know, there's viruses around. You should probably arm yourself. Like, you should try to, you know, get yourself strong. And our cells do have some antiviral things that we can do if there are viruses around. So it's just a signal that tells other cells around, you know, arm yourself, do whatever you can because there is a virus in the body. Unfortunately, that cell's going to still die because it's infected. But the hope is that the other cells won't. Growth factors just increase the rate of cell reproduction. Which again, when you're going through clonal expansion and you're making an army, you would like to make that army as fast as possible. And so it uses growth factors to increase the rate of cell replication. Our cells can secrete something called the tumor necrosis factor. This is a chemical that can recognize abnormal cells and target them for destruction. It can't kill it itself, but that's still a chemical that can almost be secreted on the outside of the cell. That's like a big, huge red flag to the immune system. That's like, hey, I'm an abnormal cell. I should probably get killed. So our immune system can recognize abnormal cells, whether they're infected with something, whether they're abnormal because they're reproducing too quickly. This is cancer. Our immune system can recognize these things and target them and kill them. And the last one, chemokine. These are chemicals that cause cells to move. The kin means to move. Kinesthetics means like movement of the body. So these are chemicals that causes cells to move. If you have an infection, if you've got a splinter and you have an infection in your finger, you need all your white blood cells to go from your different areas of your body to the finger. It's like, well, how do those white blood cells know to go to your finger? Because you got a splinter and it's infected. I'm like, there are chemical signals, chemokines, that cause white blood cells to go to where that infection is. So it causes white blood cells to move. So our cells use these different things to chat. Now, we kind of have the different kinds of T cell receptors, or the different T cells, how it works. And I've said some of this already. So our cell-mediated immune system goes against anything intracellular, which means inside the cell, and any kind of abnormal body cell. This includes cancer. The most common intracellular pathogen that it's known to find and infect are viruses. So I usually just chat about viruses, but there are parasites, there are bacteria that can get inside of cells. So other things that's affected against, yes, bacteria, cancer cells, parasites that can get inside of our cells, because we'll talk about some of those when we get to parasites. And we'll talk about some of the bacteria that like to hide out inside of our cells when we get to bacteria. But viruses really are the main target of the cell-mediated immune system. I don't know why I have these come up as separate, but this is showing an example. Here is one of our cells that's got bacteria hanging out in it. You can see the rod shapes of the bacteria. Again, our immune system, you can see that there is bacteria inside of that cell. It's going to be because it's presented on the outside, and our cells can target that cell for destruction. Now, activating your cytotoxic cells, there's really 4 steps on how we get these active cytotoxic cells to then go out and start killing all of our cells. The very first step we have is antigen presentation. So the cells aren't going to do anything. They're not going to go kill anything. They're not going to start make more of themselves, nothing, until they're first presented with that foreign object. And yes, that foreign object is going to be presented using MHC1 or MHC2, or if you're a dendritic cell, you can do it with both. It's great. It's double tasking. Now, if you're a dendritic cell, because you do have both, you're an antigen-presenting cell, so you've got, any cell has MHC1. That's going to activate our cytotoxic cells. But because it has the MHC2, it can also activate those helper T cells. And so the next part is making the helper T cells. So kind of our first step is, let's do antigen presentation. Shorten it. Then let's wake up our helper T cells, because one of the jobs with a helper T cell is to not only help the humoral branch, but it's also to help our cytotoxic cells. They're going to be better killers if the helper cells tell them to be better killers. It's great. You can also see that IL, that's some of those interleukins, just the white blood cells. There's lots of interleukins out there, but they're chatting with each other. So we're going to next wake up our T helper cells, which are going to activate our cytotoxic cells. I shouldn't say activate, because they're activated once they see the antigen, but they're going to stimulate them to start making clones. So then we're going to have cytotoxic cells making clones. So then you have this awesome army that's there to go out and kill. Now, it's going to start making more of itself, but it's not going to only make more of itself to go out and kill. In making some of the clones, it's also making some memory cells. So it starts making memory cells right away. So as it starts to make clones, this includes making memory cells. Plus memory cells. And then it will continue to keep going through that clonal expansion. Self-stimulation. Self-stimulation. More clones. So we're waking up everything. We're getting those helper T cells to start helping. We're going to make get those T cells to start making more of themselves, including some of the memory T cells, and then we just start mass producing cytotoxic cells to go out and kill. Different book, different picture showing this. This was that picture I had before, that again, when we present an antigen, which will then activate the cell, we start to go through that clonal expansion. This doesn't have the B cell involved, but we're going to make more of ourselves, including some memory cells, and then we just start going through that massive self-stimulation. We just start making more and more clones. Again, they show 4. We're talking thousands, if not millions, of these cytotoxic T cells are then going to get made and go patrol the body and target cells for destruction. So 4 steps. And I'm like, your antigen presentation, making those T helper cells because their job is to help, clonal expansion, which includes the memory cells, and then the self-stimulation to really make that mass production of all of those cytotoxic T cells. Showing some of the pictures of some of these cells at work. And I'm like, we've got a cytotoxic cell that's actually trying to kill a cancerous cell. Cancerous cells are always going to look abnormal. That's kind of the purpose of them, the point of them. They're abnormal. And they start making lots of themselves. Here's a dead cancer cell. There's your cytotoxic C cell right there trying to kill cancer cells. Now, I'm going to go off on my tangent, which I might be ahead of myself. This is the newest age of cancer treatment. We know our immune system can recognize abnormal cells and recognize those cancer cells and target them for destruction. However, although our immune system can do this, we still have cancer. So it's like, well, what happened? Well, your immune system always, if the cancer is enough, we just can't make enough cytotoxic cells. For whatever reason, it might not be recognizing one specific thing, that it doesn't even see that it's foreign, even though it is. I'm like, so there's something wrong with the system that leads to cancer. I mean, there's things that no matter what you do, if you damage your cells in your bodies enough, it can turn into cancer. And your immune system just isn't up to par to do what it needs to do. Well, what if we can make your immune system better? What if we can stimulate your immune system, to like really start beefing up these cytotoxic T cells to start killing? That's the newest age of cancer research, is literally pumping, things that increase your immune system so that your immune system can fight cancer and stop cancer. And it's working. I've actually started to see commercials now where different drug companies are, advertising like, hey, we're in the leading research of, using immunotherapy. It's using your immune system as a treatment. And it's awesome because it doesn't involve all the chemicals with chemotherapy and all the radiation, instead of let's bump up what you already have. So it's awesome. Now, those memory T cells that are made along the way can last for months, if not years, in your body. Now, months and years is different. There are certain things that, yes, your immune system can recognize, it will get rid of it, it will target, and those memory cells kind of go away after a couple months. Examples, I was going to say even COVID. I was going to say people that have COVID, can you get COVID again? Yeah, it sucks. It just means, and again, different immune systems can have different lengths on how long T cells and T memory cells hang around. But some things hang around for, 6 months, a year. It's all kind of out. They're still figuring all that out with COVID. But some things hang around for a little while, but then they start to go away. But there are some memory cells, that those memory cells can hang out for years, if not your entire lifetime. There's things that you've been vaccinated against that, if you have it once, you'll never get it again. People that have had chicken pox, it really is, once you have it, you made memory cells again, you're not going to get chicken pox again. Now, it can get reactivated and turn into something else, but we'll get to that when we get to viruses. But it's not the exact same virus doing the exact same thing it did originally. So there are certain T memory cells that last for months and some that last for years, if not your entire lifetime. And then as soon as it recognizes that same thing that initiated the whole process, they become active. So they're immediately functional upon subsequent contact with the original antigen. Whether it was a virus, whether it was a bacteria, who knows, whatever it was, as soon as they see that original antigen, they're active and they're going to go kill it before hopefully it starts to reproduce. Now, their job of a memory cell is to hopefully kill those things before you get sick. And so the secondary response, so if you ever get sick with that same thing, the idea is that it is a faster response. It can make, if it's this way, it can make antibodies a whole lot faster. If it's a cell mediated, it can make those killing cells a whole lot faster, that hopefully you can kill it before you even know you were infected. This is why even people that have gotten COVID, and I'm like, if they've got it multiple times, it usually is less severe with every subsequent infection. The again is that you've got some memory cells, some memory that you can know a little bit on how to fight things a little faster. Now that T cell regulation, as I said before, its whole job is to suppress the immune system. Regulation is needed to prevent your, I'm trying to think of the shortest way to say it, but it's really to prevent your cytotoxic cells from killing yourself. That's what we need to do. We need to suppress our immune system so your cytotoxic cells don't kill normal cells. Now, one of the ways that do that is there's usually multiple signals to stimulate the cytotoxic cells. That yes, the T helper cells can tell the cytotoxic cells to do stuff, but there's usually other signals, other interleukins and stuff that try to help trigger that as well. Slows the chance down that those cytotoxic cells are going to go kill. Now I'm going to jump back over to humoral immunity that we went over on Tuesday with the B cells, becoming plasma cells, churning out lots of antibodies. Because it can get activated with the help of T helper cells and without the help of T helper cells. So everything we talked about in this, it can do this just fine without any help from over there. And that's called T-independent antibody immunity. It means you can activate your B cells without any T cell help. It's independent of the T cells. So it can do everything. It can activate B cells without any T helper help at all. And it's great that we can do that. We don't need T helper cells to say anything. We don't need T cells and anything. It can do it on its own. Now, that just means it's going to recognize antigens without T cells. It's a fast response, as fast as it can possibly be. Again, it can take 2 weeks to turn out lots of antibodies. But it's going to turn out those antibodies. Those antibodies are going to go away. The plasma cells are going to go away. We usually don't get as many memory cells, and so there's little immunological memory. And the whole process is even slower in children. So we can do this. We can get rid of things in the body. But for long term, it's not super beneficial when it does it all by itself. Again, it's great. We can get rid of things. But really, there's not a lot of memory. However, if they start to work together, we have T helper independent humoral immune system. And I'll break that picture down. But really, we're going to activate our B cells with the help of a T helper cell. And so, yes, I know there's a lot on here. Most of it I don't care. But at the very, very top, we do have some T helper cells up there. They are going to get activated. Again, a T helper cell is not active until it's shown an antigen. And it's shown an antigen on some type of antigen-presenting cell. Even my pointer's too short. So there's an antigen-presenting cell that's activating a T helper cell. That T helper cell can turn into something known as a T helper 2 cell. And it's that T helper 2 cell that activates the B cells. So it's the that T helper 2 cell activates the B cells. They become active, they turn into plasma cells, they turn out antibodies, and they also turn out the memory cells. So lots of stuff, but all I care about is we made some T helper cells. And they're the things that are going to activate the B cells. Still going to go through the same process, still going to make plasma cells, still going to make memory cells, still going to make antibodies. It's just the T helper cell activated those B cells, not the foreign object that was floating around. Which means you can have a virus inside of our cell. That cell is presenting it to those T helper cells, and the T helper cells are going to present it to the B cells. So the B cells are like, oh, okay, so we're looking for that thing. Now, the benefit of it, so it does use antigen-presenting cells, it uses the T helper cells, and it uses our B cells. It's just word. It's the same thing that was on the other one. Present antigens, wake up the T helper cells. They make T helper 2 cells. They activate the B cells. The benefit of it is the memory B cells that are now made. These guys, when they all work together, those memory cells last longer. Because honestly, the memory cells, if we do this all by itself, maybe a couple weeks, months tops. Memory B cells that are activated when it goes through and uses both systems can last a whole lot longer. Now, memory B cells themselves don't make the antibodies. They're just there so that when they do become activated, they can make and become those plasma cells to turn out antibodies. And they become activated as soon as they see the original antigen. So whether it was a red circle or a green triangle, whatever it is, those memory B cells are going to become activated years later. And they're going to immediately start turning out antibodies. And when I say immediately, immediately is like 2 to 3 days. But it's a lot faster than 10 to 14 days, which is usually about normal. So when the two work together, you do get a faster, better response. Now, this is showing, again, here's some of my days of how long it can take for you to start making antibodies and which are some of the antibodies that you actually start making first. So the first time you're exposed to antigens, they can take a good 10 to 12 days we got to activate those B cells. They have to become plasma cells. Then they have to start turning out antibodies. And we might not even start to see antibodies in our bloodstream whatsoever until 10 to 12 days later. So again, at this point, you're probably quite sick. And the first antibody that we do start making is that antibody, or IgM, M. A little bit later, so like even a month later, we start to see some of the other antibodies getting made. But by that time, you should be feeling a lot better once we start to get these guys. So it can take, again, a couple weeks before we really start making enough antibodies to get rid of that foreign invader. However, the second time you are exposed to the same thing, so whatever it was that you were exposed to the first time, we don't have this 10 to 12 day lag, this 10 to 12 day period of waking all these cells up and making plasma cells to turn out antibodies. We have just a couple days. And we start to get huge responses of that IgM, that first one that's made, but an even bigger response of IgG. So we have this huge number of antibodies that is produced in a super short amount of time. Again, the idea is that if you get exposed to this thing a second time, you can make antibodies so fast that you don't even get sick enough that you would notice you were infected. This is the idea with any kind of vaccine, is that when you get a shot, when you get a vaccine, you're exposing yourself to it. Again, it can take weeks for you to actually make antibodies against something. But if you're ever exposed to that same thing later on, you can start to make antibodies super, super fast so that you don't get sick. Now, they also call this an anamnestic response. So if I get rid of like the AN, what is amnesia? If people suffer from amnesia, what happens to them? Yeah, they forget everything. They're like, I don't know who I am. That's amnesia, is forgetting. When I stick AN at the beginning of something or A at the beginning of a word, it means without. So without forgetting, which is the longer way of saying memory. It's like an anamnestic response. It's a not forgetting response, aka it's a memory response. And so that anytime you're exposed to something a second time, you have memory cells now that you can remember how to do something about it. You remember how to fight that war the first time, so it's a whole lot easier to do it the second time. Now, I've got this link on Blackboard, because usually I have students that are like, oh my God, this is like the worst lecture that I've ever been to. Because there was that chapter 14 lecture on the innate immune system, and then there was all these different types of T cells and B cells and different kinds of T cells. This is like a 7-minute YouTube video. I'm not going to play it right now. That's a nice summary of the adaptive immune system. It doesn't go through as much detail as we just went through, but it's a really nice summary of what's actually going on in the adaptive immune system. Now, as I said before, our whole job is to hopefully, get rid of the foreign invader, but also to make some of those memory cells so that we can get that memory response or the anamnestic response. And so there are different ways we can acquire immunity. There's naturally acquired immunity and artificially acquired immunity. A naturally acquired immunity means you actually got that pathogen. Like in your normal, everyday, going around your life, you acquired that antigen. So if you got COVID because you got COVID, like someone coughed on you, sneezed on you, whatever, that's a naturally acquired immunity. Again, it wanes. But you actually, are immune to it. If you had the flu, like you actually got the flu, and I'm like, you have a naturally acquired immunity to that particular strain of flu. Artificially acquired immunity is because you have immunity via a vaccine. It means you did not acquire that particular pathogen in your normal everyday life. You acquired it via a shot. Now, how many, I'm going to pull you, and every year I do this, my number starts to skew one way or another. And it's one of those, at some point I'm going to have no one raise their hand. But raise your hand if you have a naturally acquired immunity to chickenpox. Just one. My number gets smaller every year. So what does that mean? Yes, a nice raise hand. I had the chickenpox as a kid. It was a rite of passage. It's like my sister got it first, and my mom made my sister give me a kiss. Just so that she's like, if I have to take off work for one kid, I might as well have both kids sick. Thanks, Mom. So it's naturally acquired. It means you have actually had the chickenpox. Now, that means like the majority of you, and again, every year my number skews as you guys get younger and I get older. That means the majority of you, I assuming, have an artificially acquired immunity to chickenpox, which means you've all had the chickenpox vaccine. Now, we'll talk about chickenpox when we get to viruses, because you guys are all going into a healthcare setting at some point. Me and Diana, I'm like, I'm not going into the healthcare facility, but she is. And anyone that's actually had the chickenpox and goes into the healthcare setting is going to have a titer done. Why you're going to have a titer done. And we'll talk about titers. It really is just looking at how many antibodies do you still have? How many antibodies in your body do you still have against chickenpox? Reason for that, if you guys got a chickenpox vaccine, there should be a medical record that you had the chickenpox vaccine, so that if you're working with a patient that has chickenpox, you can prove, like, well, I've been vaccinated, I should be okay. If you've actually had the chickenpox, there is no medical record. My mom didn't take me to the doctor. You just get a horrible rash everywhere. But there's no medical record that I've had the chickenpox. Even though I can tell people, well, I've had the chickenpox, so I'm fine. I can work with that patient. They'd be like, but we don't know that. So they will actually run a titer and to look to see how many antibodies I have in my body, to see if I truly am immune to working with someone that has chickenpox. If I'm not, they're just going to give you a shot. So people that have not had the chickenpox vaccine, they're going to run a titer. They're going to see how many antibodies you have, and you may or may not just need to get a vaccine to make sure that you actually are vaccinated and have immunity to that particular virus. Now, we do call immunity, we break it down into, do you have an active immune response or do you have a passive immune response? An active immune response means that you made antibodies. Like, it was active. This whole process was active. You were exposed to something, whether it was by a vaccine or whether it was by a natural encounter with something, but you made the antibodies. Your immune system was very active and you made the antibodies. So you have antibodies in your body because you made them. That's an active immune response. You actively made them. A passive immunity means you have antibodies in your body that you didn't make. And I'll give you a couple of examples on when and how that happens. So passive is you have antibodies that you didn't make. So your immune system didn't do anything. But you have antibodies. One of the top passive immunities is mothers and breastfeeding. That milk has antibodies that mom made, but the baby now has antibodies through the breast milk. So that baby has passive immunity. They have antibodies in their body. They didn't make them. So they have some immunity. They just didn't do a thing. Another example of passive immunity is if you are given antibodies. Some of the top things that you may encounter, I'm going to give an example, but it's not a top one. So one top one is if you have a patient that has tetanus. It's pretty rare because you can get vaccinated against it, but not everyone gets vaccinated. So if you have a patient that has tetanus, and we'll talk about tetanus when we get into bacteria, but there is a toxin that's super deadly that this bacteria makes. There's another one I'm like, that's super deadly, botulism. There's a botulism toxin that if you have a patient that has tetanus or has botulism, I mean, honestly, by the time they get to the hospital exhibiting symptoms, their immune system is, it's trying to do this, but they will die before it ever gets to full activation. So what we can do is we have antibodies. And we can give antibodies to these patients. And those antibodies can bind up the toxins and neutralize them. And then while it's trying to get rid of the toxin, your immune system is still trying to get rid of the actual bacteria and any residual toxin. So we can give patients antibodies. Again, I think it was just a couple years ago, 3 years ago. It's pretty rare to have patients with tetanus these days because everyone seems to be vaccinated or enough. But it was a couple years ago that there was, I don't remember which hospital in La Crosse, but there was an Amish man, again, they don't get all their vaccines, and they work in the dirt, that came in with tetanus. I think he had to go into a coma for several months. And they did eventually give him antibodies. And I say eventually is because it's rare enough. Our local hospitals don't just carry antibodies. They have to specially order them in. And so he made it, but I think he was in the hospital for a few months before they could get the antibodies. Another example of giving antibodies, if you're bitten by a venomous snake. If you're bitten by a venomous snake, again, your immune system can recognize that venom. It can wake up your B cells. It can wake up your T cells. You can start to make antibodies. But again, it could be 12 days before you even start to see the first antibody. And are you going to be alive 10 to 12 days after you get bitten by that venomous snake? Nope. And so that's when they give them antivenom. Antivenom is antibodies against the venom. And so I'm like, that's examples of passive immunity, is when you can be given antibodies. Mm-hmm.
So why is kind of off topic, but why do people say that if you get stung by a jellyfish, you should pee on it?
That you should be fine?
No, you should pee on it.
You're not. Nope, it does nothing whatsoever. I think, I'm trying to remember because I read too, what actually happens. Because in theory, urine is sterile, that it's almost rinsing it, which would be better than ocean water. But even then, as it leaves the body, it still picks up some of your normal flora. Some people will say it lessens the sting, but there's no actual data that it does. And so I'm like, if you're bitten by a venomous jellyfish, you got to get in. You got to get antivenom. But even that, sometimes it's not fast enough by the time you get to a hospital because, it's Australia where you get the box jellies, that it's the most deadly, and then they literally come out of in barbs, and then that barbs have venom, so the barbs stay in you, so it's you can't even remove the venom. But yeah, you got to get antitoxins against that. So there are things. I just read a thing on Facebook, don't ever believe everything you read on Facebook. But it was a super interesting story about it was in this area, about horses that were dying. Did anyone see this? You know, sometimes it just came on my Facebook a couple weeks ago. There was a bunch of horses that were dying, like literally just they were fine and then 2 hours later dropped dead. And it was like multiple. And I'm like reading through this, I'm like, I don't have horses. But I was like, oh my God, what, why, like how? Eventually reading the horses, what they were eating was a food that had the botulism in it. Because when they had the food in it, an animal had gotten into the food, and botulinum grows in an anaerobic environment. And so inside that, that bale of hay was an anaerobic environment, and the animal that was in it had the bacteria, and so it was like a breeding ground for this. And all it took was enough of that toxin that they ingested, and that toxin killed them. Very, very quickly. They went from fine to they can't breathe, they drop dead. Again, we'll talk about botulism later. It's awesome. It's so cool. But my interesting fun factoid, if I take, I'm going to write, the first 2 letters of botulism, and I mix it with the first 3 letters of toxin, what do you get? Botox. Fun fact. That is what Botox stands for, is botulism toxin. That's why there's always the, and if you experience trouble breathing, blah, 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 blah. I'm like, yeah, it's bad. It is a toxin. So this is just showing some examples of some pictures. Naturally acquired immunity is because you actually get sick, like the chicken pox. This is an active, like you're actually making antibodies against it. Naturally acquired, passive, putting the two together is the mom acquired something. Now the baby gets that antibody passively, but it was still a natural acquired. Anything artificially is you're given via a shot. Whether you're given a vaccine or whether you're given an antitoxin. Like it's given via a shot is the artificial. That was the end, and we actually finished a little early. So, but we did finish it, which means what do you have to do before I see you next time? And like you'll have the quiz on it. Was there anything else? Questions 7 and 8 about like last week's material is due tomorrow as well. So if you didn't answer those microbe questions, questions 7 and 8, they're due tomorrow. And then you've got a quiz on this due Monday night by midnight. If you've got lab after this, I will see you.